Okay, so today I'm going to tell you about what we can now do with de novo protein design. And I'm going to sort of start by framing it in the context of the coronavirus problem. So the basic principle underlying protein design, well, up until very recently, is that proteins fold to their lowest free energy states. Now, this isn't always true, but it's a good, it's a good first principle for organizing thinking. So if you want to predict the structure of a protein from sequence, you search through the different possible states of the protein and evaluate their energies. And the native structure will be the lowest energy structure. If you want to design a protein, you make up a brand new structure, and then you need to find a sequence whose lowest energy structure is that state. And we've developed a computer program, we and our colleagues, called Rosetta over the years, that does the sampling in sequence space and in structure space and evaluates energies. And we've been sort of progressively increasing the Rosetta energy function, trying to describe van der Waals interactions, hydrogen bonding interactions, solvation interactions as accurately as possible. Now, very recently, this whole picture has gotten a bit upended, and we're using deep learning networks to hallucinate new proteins. And if there's time at the end, I'll say a few words about that. So what I'm going to focus on today throughout my talk is the problem of de novo protein design. And this is a very interesting problem because the number of possible sequences, even for 100 residue protein, is astronomical. And nature and the evolutionary process have only sampled a much, much smaller fraction, a very, very tiny fraction of this. So if we think about all of sequence space being represented by this gray bar, then naturally occurring proteins, fall into these clusters, protein families. And what I'm going to be telling you about today is designing new proteins from scratch, from first principles, that are completely unrelated to any naturally occurring protein. So I'm going to tell you about a lot of proteins today. None of them have any sequence relationship to any protein of known structure. And so in all of the everything I'm going to tell you about today, after we do the design calculations, we obtain synthetic genes that encode these new amino acid sequences. We put the synthetic genes into bacteria or yeast. They produce the proteins, and we purify them out and then characterize them. We're also doing a lot of work now going beyond the 20 naturally occurring proteins, 20 naturally occurring amino acids, and making synthetic molecules by chemical synthesis, but I won't be talking about that today. So I'm going to organize this around COVID. So when the pandemic broke out, as you know, the sequence was published more than in January of a year ago. And when from the sequence of the spike protein, we were able to build actually quite a reasonable model of the spike glycoprotein. And then as the cryo-EM structure and crystal structures were determined, we switched over to using that. I'm going to tell you about what we have been doing as far as antivirals, diagnostics, and vaccines. So for antivirals, we have been focusing on blocking the interaction between the spike protein and the ACE2 human host cell receptor. So we've been, it was kind of, the timing was interesting because we've been making a lot of progress recently in designing proteins to bind to protein structures of interest. So basically, without using any information other than the target structure, we can now design binding proteins. And so that's sort of depicted on the right here. So we make up very large libraries of hundreds of thousands of different protein backbones. And then we dock those against the target. In this case, this is the receptor binding domain of the virus. And we identify those docks which are shape complementary and from which we can make chemically complementary interactions with the virus. So we first dock the scaffolds and we design the interface. And we pick out those where the interactions really look optimal. So the other approach we used here was to take a piece of the ACE2 helix. So this is using kind of privileged information because we have the structure of the complex between RBD and ACE2, and then we built small proteins around it. So this one's an ACE2 mimic. These designs sort of cover different binding sites on the surface of the virus. And here are examples of proteins produced, designed using these two different methods. So this is one that was designed completely from scratch. And it's 55 amino acids, hyperstable, and it binds with about 100 picomolar affinity. Now, interestingly, the one where we use the ACE2 helix is, first of all, it's a bigger protein because we need to fit the ACE2 helix in, and it's considerably lower affinity because it's based on ACE2. It's closer to 10 nanomolar. So we can actually do better in terms of binding an arbitrary new target by building from scratch. Okay, so this is now the cryo-EM structure of this 55 residue de novo design protein bound to the spike trimer. This is from David Biesler's lab, and you can see one of the RBDs is in the downstate, two are in the upstate, and you can see the protein in ribbon here. Now, what's really exciting from the computational design point of view is if we compare the structure, the experimental structure shown here, with the computational model, which is what I described this docking and design process, you can see that the design protein is exactly where it was supposed to be. The backbone is in exactly the right place, not only sort of internally in the fold, but also placed relative to the RBD. So the computational model and the design are superimposed on the RBD, and so you can see the design, the design protein is in exactly the right place. If we zoom in and look at the side chain interactions, we see that those are nearly identical to the design model as well. So this was really our first indication that we could design, well, one of our first indications that we could really pick an arbitrary site on an arbitrary protein scaffold and design very high affinity binders to bind there. Okay, so these proteins are very potent neutralizers of the coronavirus. So this is an experiment where live virus is being added to cells, and then different designs are being titrated in. And the one that I showed you is this magenta line here at about 15 picomolar, which is 0.15 nanogram per mil. This protein gives is blocking entry. That's the IC50. And this is it's a small protein, so this corresponds to just 0.15 nanogram per mil. It's perhaps the most potent compound known to date to block entry, block the ACE2 coronavirus interaction. Now, there's been a lot of, with collaborators, we've been doing a lot of animal experiments. This is with Syrian hamsters, which are one of the classic models for coronavirus infection. So hamsters that get the virus get very sick. It's a very acute infection, and but if this protein is administered intranasally from 48 hours before to 24 hours after, there's almost no effect. You can see that we're getting nearly complete recovery by 7 or 8 days here. So far I've described, I showed you that the small protein can bind to 3 different RBDs on the coronavirus. And so we've obviously been very concerned about escape in the last 6 months. And so we focused on, even though we're at 15 picomolar binding of the individual domain, we thought we could obtain even higher avidity with higher affinity by making constructs which engage multiple RBDs simultaneously. So here what we've done is to design a trimeric version that holds 3 of these binding domains in exactly the right orientation needed to bind to engage 3 of the RBDs. And here's some negative stain EM data showing that the trimer, this design trimer, in fact, sits on top of the spike. We actually have a pretty nice cryo-EM structure of this now. So we can take binding domains and then position them quite precisely. And I'll be alluding to that in a moment about in the context of designing biased and superagonists for cellular receptors. So with these multivalent versions, both trimerized versions like the one that I just showed you, and versions where we take 3 different domains and connect them by linkers, we get very potent protection against all the escape versions of the virus that we've seen. So this is the South Africa and the UK versions, for example. And these are these different multivalent versions. We're again getting IC50s, which are better than about 100 picomolar. We're seeing similar protections in animals as well. Another nice thing about these small proteins is they're not like antibodies. The problem with antibodies, particularly for something like the coronavirus, is you just can't, there's not enough manufacturing capacity to produce them for everybody who would need them. But these small proteins express very well in E. coli. So this is just a whole cell lysate expressing that de novo design protein. It's down at the bottom, 55 amino acids here. You just heat the cells. The protein, most of the E. coli protein denatures. This is at heating at about 80 degrees. And the only thing that's left in that's soluble after spinning that heated lysate is the designed protein. There's some small levels of contaminants which can be cleared up quickly. So we think this is a way to really, really drop the cost of drugs and coronavirus antivirals in particular. So we are we're sort of going through the steps to get these to clinical trials, and that's been a very interesting. There aren't one of the tricky things has been that there isn't a lot of precedent for the idea is a nasal spray, so you could just have it. You don't have to refrigerate this stuff, so you could just have it in your go to the drugstore, get it sprayed in your nose if you're going to get on a plane, or if you're in India and just in a dangerous place, you could take it when you needed it. And again, it's super stable, so you won't have to keep it refrigerated, and distributing it should be much more easier. And then the other thing that we're working about on this context is it did take us a while, and there was a fair amount of experimental screening that we had to do to find these really, really good designs. So we're trying to now improve this whole computational pipeline so we can go from sort of identification of new pandemic threat to very high affinity binding proteins in just a few weeks, which we think is possible. Now, we've taken the same methodology, or actually in parallel, Longjin Cao and Brian Coventry really made this enormous breakthrough in the last several years, being able to sort of, like I said, take a protein of known structure, pick a region, and then design a very small protein, less than 60 amino acid, that binds to it. And so they've been making binders to the major cell surface receptors like FGF receptor, PDGF receptor, EGF receptor, and then many cytokine receptors. And I'm just showing you a few of them here. And we have crystal structures now, many of them, and again, they're binding very much like they were supposed to. And these are all, it's kind of interesting, they're all very small proteins. This just shows surface views. And they have their shapes and chemical properties, though, are different enough to make them very specific. So this is the different design binders, looking at binding TED experiments against the different targets, and you can see we're getting really good specificity here. In the case of the EGF receptor, we made binders to different receptor subunits. So now what we're very excited about doing is trying to manipulate cell fate by making super agonists and different types of agonists which hold different receptor subunits together in different orientations to get new signaling outputs. And we have there's a first example of this. Where this is from a year or two ago with Chris Garcia, where basically we made scaffolds which held the Perutz receptor at different angles and distances from each other. And what we found is we got different signaling outputs. So now with these small binding domains, we think we can really generalize this to signaling. There's all sorts of different exciting applications like trans-differentiating cells in the body for regenerative medicine and other applications. So very excited about that area. Okay, the second area, in response to COVID was diagnostics, developing sensors. So far I've talked about designing these really rigid sort of rock-like binding modules. But for this, we wanted to see if we could build molecular devices. So here we have a molecular device which has two states, has a closed state, which is dark, and an open state, which emits light. And these this opening closing transition is coupled to the free energy of binding a target. So we've got a binding domain that's closed here, and when the target's present, the binding energy drives opening. And so what we've done here is simply take that small coronavirus binder that I described, embed it in the closed state of the system, so that then when the coronavirus is there, it opens up and light is reconstituted, light is emitted. Basically, luciferase gets reconstituted. And this works really well. So the sensor is dark, and then when the spike or the RBD is added, we get this very rapid and large increase in luminescence.